Hi everyone, welcome to today's seminar. Today's speaker is Letizia Campioni, and I would say just kick it off.
Okay. Well, thank you very much for coming. And before diving into the core of this seminar, I take a moment to introduce myself. There are many new faces for me in this audience. The last time that I made a presentation here in the EBD, it was in 2012 when I defended my PhD thesis on the movement ecology of eagle owl. And after that, I moved to Portugal with a postdoc fellowship where I started to study the ontogeny of migratory behaviors, development of trophic niche and dispersal in different species of seabirds. And after that, I had the opportunity to stay there for more 6 years with a research contract. And during this time, I kept working on my research interest. But at the same time, I started to expand my research line, and I started to work with seabird conservation, pollution in general, and interaction of seabirds with human activity offshore, and also ecotoxicology. And are actually this topic that brought me from Atlantic ecosystem to the Mediterranean, because thanks to this overlap with my interests and the ones of Manuela Forero, she contracted me within the context of the PCBO project. And since February, I'm here again in Doñana. So, saying that, I think that we can move on with the seminars that is structured in this way. Well, I would like to start with making the point of the situation of their major threats, and particularly with a focus on small petrels. After that, I will move with you. We will travel to the Bermuda archipelago, and here I will talk medium-sized petrel that is endangered. And so we will start from the past to understand current population status. And then the final part is related to the work that we did to enhance the conservation and the knowledge on this poorly studied species. Everyone knows that we are in a time of global conservation crisis, and seabirds, unfortunately, are one of the most endangered group of birds at global level. So what we know is that more than 50% of seabirds for which we have population trends are in decline. And when we go and look to the threats that are impacting this population, a recent review published by some colleagues highlighted that more than 80% of seabird species are threatened by one threat, 70% by two, and more than a half by three. So the panorama is not very nice for them. And when we go to look into the threats, of course, we have a top list, and at the top of this list, there are bycatch, invasive species, climate change, and overfishing. And if we look to some more threatened groups, taxonomic group, we can appreciate that different taxonomic group is affected by different types. But today we will not talk about albatrosses and penguins that I know are other species, but we will focus on less popular small petrels. So within this group of petrels, there is a genus of the Pterodroma petrels that include 34 species. They are globally distributed, and they share several ecological and behavioral characteristics. One of these, the most intriguing one, is indicated in its genus name. So Pterodromas, wing and run. So they are highly adapted to prolonged flights. They are able to select winds to optimize their flights and distances at very low energetic expenditure. But despite the growing interest in studying this group in recent years, well, when we compare what is known about the ecology and behavior with larger seabirds like albatrosses or penguins, well, they are still poorly understood. And in addition, also their conservation status is not very good, because overall of these 34 species, more than 70% has a compromised conservation status. One is extinct. So, well, we are quite worried about this group of species. And look to the major threats to this group of small petrels. Well, this histogram tells us clearly that major threats are on land when these birds come offshore to breed. And in the marine environment, climate change and severe weather apparently seems to be the only problem for them. But when I look at this histogram, the first question that came to my mind was, well, it's true that the oceanic environment is really a safe space for them, or we don't have enough information to make a proper assessment. So along this seminar, I will try to give you an answer. But to do that, of course, we cannot explore all the species, but we come back to our latitudes. And it's worth to know that in the North Atlantic, we have 5 Pterodroma species. Each one is endemic to each oceanic ecosystem. And today we will go to the Bermuda, where we have the Bermuda petrels. As I told you before, it's an endangered species. And well, sometimes it's called also cahow. That is how it's popular on the island. And its population size is quite small, because currently we have more or less 165 breeding pairs. Well, to understand how this population get to this small number, as I told you, we have stepped back into the past. And until the 16th century, when Bermuda were discovered, and since then, the island was used like a stop-off place to refill with food from sailors that from Europe were crossing the Atlantic and reaching the America. Well, at that time, Bermuda was like a heaven, full of biodiversity, animals. And so Spanish people were among the first to touch this island. They called them the islands of devils. They were really scared by them, by the island, by their dangerous reef, but also by really weird things happening at night, where they start hearing creepy sounds like children crying all around. And so legends say that's the belief that the island was full of devils. They were looking for revenge. But actually, they were the Bermuda petrels that were coming ashore at night, and with their crying vocalization were, well, creating this weird atmosphere. So the only people that settled down in the island, that it was in the 17th century, were British people. British people settled down there. They then bring a lot of domestic animals, invasive species, and what they used to do was to collect the eggs, to catch the bird, put under salt, and use as a food for their traveling. And so in less than 20 years, the species was officially declared extinct. And this belief lasted more than 350, more or less. When in 1951, this group of American naturalists went to Bermuda with the idea that the species was still alive. And they were searching for it in very small outer rocky islets that were in the outer side of the reef. And they found them. They found a small number of breeding pairs in small crevices. And so the government of Bermuda, a few years later, started a recovery program that was led initially by David Wingate, that was the terrestrial senior officer of the Department of Bermuda. And after 40 years, this title moved to Jeremy Madeiro, that is the current conservation officer in charge of the population, the management of the population, and the guy that we collaborate with. So what were the problems that these people were dealing at that time, but over more than 60 years? First of all, the strong anthropogenic pressure. Bermuda is quite small. It's more or less 50 kilometers, square kilometers. Population in 70,000 individuals. So population density is very high, but it's even worse when it's the touristing season, because Bermuda is a magic place where, our major income of this country is tourism, and number of people that are on the island, it's 10 times higher than the resident population. So the space available for this species is very restricted to this northeast area of the Meta, has this hook shape. And actually it's in Castle Harbor Nature Reserve that is protected. It's also an ABA. The birds were rediscovered on these 4 small islands in red, but actually there is a colony, 2 colonies also on Nonsuch Island that I will talk about it later. And we also know that when humans are coming in a place, they are bringing with them animals. And so actually there is a rat control program going on. There are no cats on the breeding island. There are also natural predators, but also there are nesting competitors that can also kill the chicks and occupy the nest. In addition to this, Bermuda is in the northwest Atlantic, 800 kilometers from New York and from Boston. And this area is highly impacted by hurricanes and tropical storms. And we can see that over time, the frequency of these storms are increasing. And unfortunately, these are real pictures from there. This is one of the breeding islands. And they are overlapping with the time in which these birds are coming back to mate, and so entering their nest. These are some of the nesting islands. So you can see that they are not very high to the sea level. And they are made of sandstone that is very easy to erode by wind, by waves. And so the islands are fragile and they are collapsing little by little. And this is causing a loss of habitat. And indeed, to try to buffer this problem, what they did was to build artificial nests made of concrete. They look like mushrooms, and they are underground because the species with a tunnel of connection that has an L shape to avoid that when water is coming is not getting inside the main chamber of the nest. Well, those solutions were not solving the problem. And so what Jeremy is starting to do since 2004 is to translocate the birds. He went to Australia, learned how these techniques. And so he started to translocate birds from the more exposed islands to Nonsuch Island. And now there are 2 very good colonies that are growing, and the second generation of the first birds that were translocated are already started to breed. So overall, well, to summarize, 60 years of wars with only 1 plot, it's not a lot, but finally at this time the population is growing steadily, but this is also due to the fact that the species, like many procellariforms, have a very slow pace of life, so they only produce 1 egg for breeding season, and the chicks, the birds are starting to reproduce at the average age of 4 years. So everything go very, very slow. And so from 2019, we step in. We contacted Jeremy Madeiros, and we started a collaboration. And I can see that we started to write a new chapter of the species. And this was thanks to funding that we got with these 2 projects that enable us to integrate the nesting habitat restoration they were doing with our skills in research and establishing an individual-based biomonitoring program at sea. What we did together was to identify major research and conservation priorities. The most important, not all of them, are the ones that I'm presenting here. So first of all, we had to characterize the distribution. We have to know where the species is going to understand what could be their threats. And so we characterized the oceanic distribution. We studied their foraging strategies, diets, to understand the ecological role of these birds in the marine ecosystem. Being an endangered species means that we have to also try to enhance the legal protection at sea and identify their interaction with potential human activity. And what to say, the Bermuda, it's a protected species in Bermuda. It is protected in the US, but it was not in Canada. And here is where we started to work with Canadian people. And currently the species is now under evaluation to be included in the protected species, non-breeding species of Canada. Also the population is so small that to recover needs to really reproduce with a very good success. And so we decided to examine in depth, identify potential causes of failure. We accomplished with most of this work. Many studies and activity are still ongoing. And we have a PhD thesis that is starting next January. Today I think that I will be able only to focus on these third goals and show you our results. But I'm available to prepare another presentation seminar if you are interested. So what we did was, at the end of each breeding season, Jeremy was producing reports of the monitoring of the population. And so we could evaluate the breeding success over the last decade. That was not very high, at least for seabird population. And we identified that breeding failure was occurring primarily at the stage of incubation and hatching. And so we wanted to understand why. So we thought about generating 2 possible hypotheses, non-mutually exclusive. The first one was related to the role of genetic factors that could be reducing the genetic compatibility between mates because of the bottleneck that for which we have historical information. And so this could lead a reduction in hatching success, maybe through an early death of embryo in egg. The second hypothesis was much more related to interaction with environmental factors, so exposure to contamination. We know that there are several contaminants that are toxic for during the development of embryos, and also in this case we thought that could be an additional explanation. And so, first of all, we explore the hypothesis 1. In this study, we evaluated the genetic diversity, inbreeding levels, and their effect on individual fitness, in this case, the trait that we consider was hatching success. We evaluated 31 breeding pairs, male and females. And among our major results, well, we identified the genetic signature indicating that the population suffered a recent genetic bottleneck. We evaluated their mitochondrial and nuclear diversity. Well, the mitochondrial diversity was a bit low compared to other species, but the nuclear one was not. And when we assessed the relationship between genetic diversity and fitness, we found no evidence that inbreeding or relatedness between mates affect hatching success. And so we switched to the second hypothesis related to the effect of environmental variables to contamination, and we had evidence supporting this hypothesis because David Wingate, the first senior officers, published a paper in Science in which he was describing the strong decrease in hatching success during the decades of 50 and 60, and contaminant analysis they did on eggs and chicks. And so he speculated that this relationship was real cause and effect. And this is, well, probably some of you know that at that time US was misusing pesticide, the popular DDT. The tone of this pesticide was entering any type of ecosystems, and the Bermuda petrel was very likely affected by DDT impact. Well, most in general, what happened to a contaminant that is persistent in the environment and with some specific chemical-physical characteristics that it can bioaccumulate in long-lived species. So what happened is that the contaminants, long-lived species like petrels, will be exposed to for a longer period of time, a lifetime, and it also can biomagnified along the food webs with higher trophic level predators like seabirds can be exposed to a higher level of this kind of contaminants. So we selected a group of well-studied contaminants from different origin, pesticide from the industry, others originated from human activities. All of them are persistent organic pollutants except mercury, that's it's an inorganic toxic element. And its toxicity, it's when it appears in its methyl mercury compound, so when it is methylated, because mercury alone is not so toxic like these compounds. These are the number of different type of compounds for each categories, but what all these contaminants have in common is that they effect on human and wildlife health are known, so they can cause acute or chronic effects at individual level with consequences on population. One of the most known effect is impairing reproduction. Most of them are toxic for the embryo, but also can interfere with physiological function, hormones, disrupting behaviors, and many other effects. So we do different studies. One, well, that is already published, in which we evaluated POP, the persistent organic pollutants. We analyzed their concentration in bloods, and then when we related it to the hatching success, we consider only females, using like a mechanism of transfer, the maternal transfers. Well, during the production of the eggs, female can put in movements from different tissues, contaminants that are stored, and then transfer them to the egg. The second study is based on the same reasoning, but in this case we evaluated the levels of mercury in their body feathers during the non-breeding season, that is when these birds are molting their feather. And we also evaluated the link with hatching success. So if we go to the results of the first study, well, what we can appreciate is that Bermuda petrel are contaminated. We found all groups of contaminant except one group, that is the ones related to particular using like flame retardant. And so we found pesticide, PCB that are, well, quite popular, and PAH. So in a previous study, we analyzed the diet of the species. Bermuda petrel feed exclusively on mesopelagic prey, so deep sea prey, living at between 200 and 800 meters below the water surface. So it's belonged to the deep sea food web. So what we can highlight is that also very remote places, pristine area or oceanic region are all contaminated. What dominated the contaminants profile of the species were PCB. We analyzed 55 contaminants. We found 33, and 9 of them were in more than 50% of our sample. We don't have baseline level to compare with, because it was the first study that was evaluating this, their contaminants profile. But we have that work published by Wingate in the 60s, and only for DDT. So I know we are comparing different biological matrix, so the comparison is not really comparable, but in general we can say that current concentration are 1,000 times lower than in the past. We did not find any difference in bioaccumulation pattern between female and males for the different group of organic contaminants, but PCB seems to be bioaccumulated according to the age of the birds. So older birds have significantly higher concentration compared to younger birds. And when I say younger, they are always breeding birds, not chicks. Well, when we finally assess the relationship within a hatching probability and the concentration in blood in females, well, it came out that hatching probability was lower in females with higher blood PCB concentration. Moving to the second study about mercury. Also in this case, our birds were contaminated with mercury. They were exposed. This is something that is quite common in seabirds, particularly in those feeding on fishes. Levels are relatively high compared to other species. We did not find any patterns of bioaccumulation between male and females. And also birds of different ages have quite a comparable concentration. How should we putting things in a context? So the Bermuda petrel is how it's doing with this mercury concentration. So comparing with other Pterodroma species, the cahow is at this point in a scale built by Ackerman, in which each presents the risk of observing adverse effect of mercury on reproduction, health, behaviors. So we could say that it seems that we are in the low area of moderate risk. But then again, we went to check the link between hatching probability and mercury concentration. And again, we found a significant relationship where predicted hatching probability was lower in female with higher mercury concentration. Well, this is not easy to disentangle because seabird species respond in a different way and very specific way to different mercury concentration. Well, for instance, the higher value found in seabird was in a wandering albatross that was 95 microgram per gram, and our average value are around 14, but they did not serve any impact on fitness. So it's something that probably should be evaluated combining baseline level with testing their effect on different response in health, reproduction, or behavior. So, well, take-home message from our work. Well, at sea biomonitoring actually proven to be key to fill the gap of knowledge on the behavior, ecology of the species that unfortunately I could not show you these results. It provided new insight on the existence of threat that was not reported, chemical contamination as a very important threat to small petrels. And probably when we will try to model and build some model on the demolition, we have to take into account sublethal effect of contamination on survival and fitness. And what we would like to push is that to integrate conservation program with toxicological monitoring, because particularly when we are dealing with highly mobile species, particularly if they are endangered and population in a small number. All this, our work, it was possible thanks to the collaboration with other people from different countries, students, volunteers, and well, we still have a lot of work to do. And then thank you very much for staying here and hearing what I was trying to tell you.
Thank you very much for this great talk.
Thank you to you.
Do we have questions from the audience?
Hi, thank you very much for the lovely talk. I have two questions, just small ones, really. So first, I may probably misunderstood. When do they start reproducing? How? At what age?
Average age of
4. 4. Okay, good. I thought I understood 14. I said, oh my God, this is like wow. Okay, 4. Okay, good. And second question. So, are you planning on doing then a demographic population viability model of the species?
We are planning to do. The point is that, well, you more or less saw the age of the guy that is in charge. So it's way of collecting the data. So we have books, picture of books in which he took all the notes year by year. So we need volunteers that are going to translate this information in a Excel sheet and start to. But yeah, we have information at individual level since 2002. That is when they started to ring the birds. Because the other guy, Wingate, David, the first one, he was not touching the bird. So their way of monitoring the population, they want to disturb the population at all. So he was just watching to the recording the breeding success and everything. So we have individual based information starting from 2002 to today.
That's pretty nice. Okay, thank you.
Any more questions?
Thanks for a really interesting talk. My question is also quite small, but I was curious about the mesopelagic fish that make up their diet. Does that mean they forage at night? Or are they making really deep dives?
Yeah, when I put some slides at the end because, Manuela already told me to add some of this information, but I had no time to. I thought it was too large. Well, we try to analyze their diet. We hardly collected regurgitate and feces. So we analyzed just 2 of them, and in these 2 we found 16 species of fish, 7 species of squids. All of them were of the mesopelagic but pelagic area. So they are feeding of these small fish that are like lanternfish. And well, these species, these organisms, what they do is living at the bottom of the water column, and they do daily vertical migration. So they come to the surface of the water during dark time, and we think that is when the birds are probably catching them. But well, it was strange because, well, what we see is that the birds are moving all the day around, so also during the day. And this is not a species that gets to a specific place to forage, but is foraging on the way. And so, well, we were not sure of what their ability of diving as well. So in another work that we published this year, we also use accelerometer and TDR. And well, we analyzed more than 2 million attempts of diving, and 99.99% were within the first 10 centimeters of the water. So they are not very capable of as a technique. So we are now almost sure that they have to wait and feed probably mostly by night.
So then I have a follow-up question.
Yes.
When they're at Bermuda, is that when they're nesting? Are they?
Yes.
So the rest of the year they are out in the basin.
Yes.
Okay.
Yes.
Okay, that makes more sense to me.
Yeah.
Okay.
So this is Bermuda, and these are the foraging trip during breeding. So they are, well, I told you they really cover large distances. So they forage more than 3,000 kilometers from the nest, and they maximizing, just in Bermuda during breeding.
How long is the breeding season?
Quite long, because they start to come back for mating in end of October. They stay there for months, then they leave the nest for what in seabirds is called exodus. So female go outside to feed a lot to produce the egg, and they come back early January, and then the chicks leave the nest in mid-June.
Okay, thank you.
Yeah.
So you've tested the effect of the pollutants on hatching success.
Yes.
Did you test or will you test the effect on movement behavior and this kind of?
No, we didn't.
It would be really cool.
Yeah, it would be. It would be cool. Well, we didn't because data collection is not really easy. So sometimes you go and you don't find your bird. Sometimes you lose the GPS. Sometimes you wanted to target specific individuals and the weather is not. But there is a things that I don't have any slide. We analyzed the isotopic niche of these species and it's very, very small. We analyzed in different period and within the same year, the birds seem the same, well, within the same breeding season, they seem to rely on quite similar prey. So I'm not sure that I could find very different value in mercury just because they go in different places. Because overall, this area is really, it doesn't look like, but this area is quite huge.
Cool, thanks.
Thank you. Thank you for your talk. It's not really a question, more a suggestion, but did you
Yes.
consider, because you said you had a lot of field notebooks to digitize. Did you consider a citizen science project to do that?
No, I did not so far, but it could be.
Because I was thinking on a platform like Zooniverse, you could have a nice project maybe for that. So
Well, it might be. Okay, I will think about it. Thank you.
Do we have any more questions?
Hello, I
Hi.
wonder, did you find anything unexpected in their regurgitations, just like plastics or plant seeds? Because it's quite common in their pellets.
Yeah. Well, no, we did not started any study on plastic, but well, from in the nest you never find plastic, for example. We collected few regurgitates, so in at least macroplastic. We never found or recorded any dead individual in the colony. So in this case, we could not evaluate if what was the reason of the death. Might be plastic, but honestly, I don't know. I'm not very convinced that if they feed at night. So I guess that I honestly think that most of the species of prey species they have bioluminescence. So I think that they are looking for things that are quite evident in the environment, and plastic at night is not. But yeah, if we have the opportunity, we could do some plastic analysis as well.
Okay, thank you.
Thank you.
Okay. Any more questions? I will check if there's anything online. That's not the case.
What?
Yeah.
I have another one. If it's
Yeah.
super. No, because you did a great job of introducing a species, and it's really amazing what you've been, you know, the managers and then your project have been able to do. I think this is really great. And just again a curiosity question. So what
Yes.
is it that ultimately kills them now? Like, what is their main threat now? Because you said that there has been a lot of efforts to eradicate rats. There's no more cats. So is it then maybe perhaps their survival in the off-breeding season when they go to the US and Canada, or what is it?
Well, I think the major threats now it's still on the island, on short availability of nesting place.
Okay. Oh, okay.
So human pressure.
Okay.
Because, well, there are no other places now where the population can expand, if not the islands that are there. And since they are collapsing because of the erosion, at some point, even though the population can double in numbers, it will get to a point that there will be no space. And climate change is really starting to impact. Well, the weak point of all this conservation effort, we talk many times with my colleagues, is that it seems to be based on the work of one guy.
Yeah.
So the first 40 years was David Wingate, now it's Jeremy Madeiro. So, well, what happens if nobody is going to take place?
But there is no general interest in the Bermuda population or other agencies to protect the species or to, I don't know, create new habitat even?
Well, they have interest. The government has interest until the point that this thing is not compromising other things.
Yeah.
Tourism, money.
Yeah.
And there are many businessmen that are asking her to build resorts and other places. But there is a very good community of bird watcher, and there are one association dedicated to birds. So there is a group of people that are interested. It's a national bird, so it's popular, but yeah, it depends.
Okay, thank you.
From the rewards.
Okay.
I have one question. I noticed that the girls has the foraging behavior is quite, the foraging distance and duration is very long and large, and I wonder is only the breeding individual or is just the non-breeding individual during the breeding season?
Yeah. Well, during the breeding season, birds use mostly the western North Atlantic, but during the wintering season or non-breeding season, part of the population is migrating to the east side, so they cross the Atlantic middle ridge and they get below Iceland, sometimes offshore Ireland. So there is, they are using the entire Atlantic. And now we have a work of a master student, because since there are birds that seems to be more resident in the area during wintering and other ones migrating, trying to understand what are the drivers of this in behavior. And in this case, yes, what Duarte was suggesting, we might try to understand if the exposure to contamination or other threats, interaction with human activity, could be different from these two subgroup of the population.
Okay, thank you. And then another question is, how long will the breeding individual take the foraging trip during the hatching period, or So, well, the average age of the duration of the trip during incubation is 11 days. This I just reported one record of more than 2 weeks. And during the chick rearing is between 7 and 8 days. So once they will leave their nest for 11 days, or
can you repeat it, please? No, don't worry.
For the long foraging
trip
behavior, how long will they leave their nest? Leave their eggs inside the nest?
Yes, there are, male and female share similar tasks in reproduction. So during incubation, there is one of the two in the nest and the fasting in the nest, the other one outside. And when they have the chicks, they are alternating to feed the chick.
Oh, okay, thank you.
All right. Then I have an announcement, because on Monday we have an additional seminar, so same place, same time. No seminar on Thursday, because this is apparently discussion group week, but another seminar on Monday. Hope to see you all there. Thank you again for your talk.
Thank you to you.
And thanks for attending.